my postdoc advisor, Michael Cohen, he saw this paper in 2012. This was the first time, CRISPR broke through into the mainstream. It was a science paper from Jennifer Doudna's lab at UC Berkeley. And he called pretty much an emergency journal club. I remember kind of the moment to this day where he waved this paper around and said, "We got to meet after lunch to discuss this paper." So to him, it was a breakthrough, but I had never heard of it as a neuroscientist. So we can get into a little bit more about what that paper had, but that was kind of where I first learned about it and applied it to my research.
Hi everyone, welcome to the Trial Talks Inc. podcast. I'm your host, Naomi. In today's episode, we delve into the fascinating world of CRISPR gene editing with our expert guest, Jeffrey Huang. Jeffrey holds a PhD in neuroscience from the Rosalind Franklin University, where he began his career researching Alzheimer's disease, specifically with reducing beta amyloid plaques. His scientific journey led him to a postdoctorate fellowship at the Oregon Health and Science University, where his exposure to CRISPR technology shifted the trajectory of his career. This breakthrough introduced him to applications of CRISPR in modeling rare pediatric diseases at the Children's Hospital of Orange County, and later to his current role at Mammoth Biosciences, where he focuses on advancing RNA technologies. Today's discussion delves deep into CRISPR's potential to revolutionize the treatment of genetic diseases and the transition from benchtop research to real-world solutions. Thank you, Jeffrey, for joining me on my podcast today. I'm so excited to get the chance to speak with you. So to kick it off, would you be able to explain just a little bit about yourself, your background, and how you got into CRISPR gene editing?
Yeah, thank you for the opportunity, Naomi. I'm really excited to share about an exciting tool and technology that I've been working on in research for a while now. So, yeah, just to give you a little background about myself, I received my doctorate PhD in neuroscience from Rosalind Franklin University out in Chicago, Illinois. There, I did some work on Alzheimer's disease gene therapy, basically just looking for a gene that can improve the memory and cognition by reducing beta amyloid plaques. So that in a nutshell was what I did. I mean, that's 5-6 years of work boiled down to a couple sentences. But I realized there's still a lot we don't understand about the gene therapy field, some limitations of what can be done. So that brought me next to my postdoc, or we call it postdoctoral training. A lot of researchers, after obtaining their PhD, they go into, it's almost like in med school where you have this training period. You learn how to write grants if you're in academia. And there's also love to talk more about potential fellowships that you can work on in industry. I didn't know about these at the time, but it's another avenue you can do. So I did a postdoctoral training fellowship in an academic institution. This was at Oregon Health and Science University in Portland, Oregon. It was under a physiology and pharmacology department. This was where I learned about CRISPR, actually. So my postdoc advisor, Michael Cohen, he saw this paper in 2012. This was the first time, CRISPR broke through into the mainstream. It was a science paper from Jennifer Doudna's lab at UC Berkeley. And he called pretty much an emergency journal club. I remember kind of the moment to this day where he waved this paper around and said, "We got to meet after lunch to discuss this paper." So to him, it was a breakthrough, but I had never heard of it as a neuroscientist. So we can get into a little bit more about what that paper had, but that was kind of where I first learned about it and applied it to my research. Fast forward with that. Oh, and then maybe I could pause at the end for some questions about kind of my career path, but I'll just wrap it up with how I got to where I am today. So after OHSU, I went to Children's Hospital in Orange County, where I met you, Naomi. And there I learned about, this brought me closer to the bedside part of the bench to bedside transition in research. And there I got to meet rare disease patients and their families, and it gave my research a sense of purpose, working with Dr. Raymond Wong and the clinical research coordinators, everyone that kind of carried out these clinical trials on site at CHOC. So it kind of gave me a purpose to the research I'm doing, and I was able to apply CRISPR to model rare pediatric diseases and think about ways to treat and correct those mutations. After CHOC, I pivoted to industry, where I headed up to Seattle, Washington to work at a RNA therapeutics company known as Shape Therapeutics. There I worked on neurodegenerative targets, and things are going well with that company and its collaborations.
Awesome.
Yeah, and I ended up here at Mammoth, started, I think September of last year, and really looking forward to move my learnings onto these exciting technologies that they're developing here.
Yeah, for sure. There's a lot happening in the field, and like you mentioned, you said there's a paper that came out in 2012 with Jennifer Doudna, and she ended up winning a Nobel Peace Prize just was it 4 years ago, I think in 2020 for her work in the chemistry field. So are you able to explain a little bit more about like what is CRISPR to those that may have never really heard about it, or just they have heard about it but they just don't understand the details around it?
Yeah, I'd love to get more into the basics. So CRISPR, it's an acronym, so CRISPR. I'm never going to spell it out again. I'm just going to do it this one time because it's a mouthful. It stands for clustered, regularly interspaced, short palindromic repeats. So you can understand why nobody says the full, what it stands for.
So long.
CRISPR rolls off the tongue a lot easier. And if you really dive into what each of those words means, it's essentially what it is: short palindromic repeats at the end. That means they're repetitive sequences. Palindromes, like just as race car, the word race car is a palindrome. Same thing in the genetic code. You read it backwards and forwards, it's the same. They're relatively short, so 20 nucleotides or so. And the first part, clustered, regularly interspaced, this gets at the biology behind it. The spacing is important because I'll just kind of summarize this very high level. What the intention of CRISPR was through evolutionary biology was to serve as an immune system for single-celled organisms like bacteria. So let me give you an analogy. So for multi-celled organisms like us as humans, we're able to have adaptive immunity, meaning we can, get the cold virus, get the flu virus, COVID virus, and we have these immune cells in our bodies that are able to recognize and fight that off if we survive, right? So that's that adaptive immunity. Because we have so many cells in our body, we have cells dedicated to fight off another infection of the same viral strain. In bacteria, they can't do that. They're one cell, right? So naturally, they need to have a different system, and it's a very ingenious system that they created. They've created a memory bank with these kind of sections of DNA or RNA that are like wanted posters to say, hey, like we killed off this virus that infected us. If it ever shows up again, we have the sequence, and then we can recognize it and cut it using these Cas nucleases. So that's their single cell system to fight off the same infection from that virus.
Awesome. Yeah, so we're talking about like single-celled organisms, and how does that translate into like the human body, and how we can use this to fight off diseases or change the genome?
Yeah, so it sounds like, and it was quite a leap in the application part to go from this esoteric, meaning very, unique to the bacterial viral world, and to bring it all the way to how it functions in humans. And that bridge was what, the bridge that was created, that's the work that was worthy of a Nobel Prize from Jennifer Doudna and Emmanuel Charpentier. What they did was they realized they could repurpose the system, the components, and we can get into that a little bit more. There's simply a RNA sequence and a protein that comes from the bacteria known as Cas, or CRISPR-associated protein. And you put these two pieces together with any sort of sequence, whether it's bacteria, human, you name it, animal, frog, cat, dog, anything. They're able to see that this system doesn't just work on viral bacteria sequences. It works on any sequence you create, synthetically recombinant proteins. It was just very multifaceted, almost like a Swiss Army knife where you can plug and play different sequences to target mutations and just unique sequences throughout our genomes.
Awesome. So you mentioned a little bit about like, what it is that like CRISPR is, but are there like what are specific components to this technology?
Yeah, so when it gets down to it, the two components are an RNA sequence. This is made up of a few parts. Well, and this is kind of your barcode. So if you go to the supermarket, usually you scan an item and it has a unique barcode, right, to tell you what that item is. Same thing for this RNA. It's kind of your tracking number, your barcode to direct this whole complex to a specific part of your genome. The other part is your molecular scissors. That's the Cas nuclease. So the Cas nuclease combines with an RNA, the barcode, and at that site, depending on the Cas nuclease, it makes a double-stranded cut, and this creates a break in your genomic sequence. Now, just the regular flavor of CRISPR-Cas, it creates this break, and your body's natural repair machinery comes and does a pretty, I guess it's a quick fix or patching of that region. So usually you get all sorts of insertions and deletions, and that usually disrupts the sequence. So a traditional CRISPR-Cas cut is really good at knocking out a gene, making it no longer functional. So you can see the applications are limited there because this would only apply to diseases where you have too much of a gene, like potentially like oncogene or a mutated gene that produces too much of something you don't want.
That's interesting. So, when we talk about diseases that are gene specific, that's how it gets into how we can do more personalized medicine. Is that right?
That's right. That's right. So these tools have evolved since their initial discovery or application to mammalian cells and gene sequences in the early 2010s. Now, a lot of research has been done to refine this technology, make it less like blunt scissors and more like a scalpel, where you can edit with more precision. So there's some tools now that we may get into. You have these base editors, you have these, we call them retro or prime editors, retro transcriptase editors. And yeah, it's all about getting the technology delivered to the patient's cells as well. That's another key factor, the delivery. How do we get the good payload, these technologies, to the patient's cells in need?
Yeah. So how does this technology differ from other sort of like genetic engineering that's been in the past that we've had in the past?
Right. So there's two more legacy systems, and I say legacy because these were early, they really pushed the envelope of genome editing. These were zinc finger nucleases, or ZFNs, and TALENs, or TALEN. And these technologies required modular, like protein pieces to be synthesized. And these all had to be unique. In the case of zinc finger nucleases, you have one protein with 3 nucleotides it recognizes. So if you want to look at a stretch of 20-ish nucleotides, you need to create 7 unique proteins. And the reason why that's prohibitive is each of these proteins requires a lot of money to produce. And if you're going to tailor it to each person's genome, it becomes cost prohibitive. It's basically you're making 7 proteins for each patient or, set of mutations. So, it's a great research tool, but the translatability of it it didn't make sense. Also, it has more off targets because each of these are 3 nucleotide sequences that they recognize. The chance to recognize a sequence you don't want is higher. So those are 2 drawbacks, but there are great systems that pave the way for some early studies, especially in the rare pediatric field. There are a couple diseases that were worked on as proof of concept that genome editing worked.
Awesome. Yeah, it's interesting how we're in like the cutting edge of this technology, right? Like we're just now starting to see this being used for diseases. And so it's exciting the potential for this to be used, for, like you mentioned, rare diseases, but there's a lot of different diseases out there where, we understand what genes contribute to it. And yeah, this has potential to save a lot of lives, I think.
Yeah, agreed. And one thing to note is this is still, in the grand scheme of things, still a relatively young technology. So, like I mentioned, the 2012 paper, it's been about 12 years since that initial discovery in a academic lab. And the amount of progress that's been done in just a little over a decade, it's amazing how much work has been done to push this technology forward and to try to think of ways that we can go from, you know, restoring or correcting mutations outside of the body and trying to get that therapeutic into the body. And we can get into that more, how the first FDA approved drugs for CRISPR, like these are paving the way for how we're going to go about more less tractable, more difficult to treat diseases in the disease tissue of patients.
Yeah. Yeah. Awesome. Yeah. So, when we talk about lab research, I think there's because we focus on the clinical trials mostly, at least that's the audience that this is like targeting. But like what
Yeah.
goes into play on the lab research side with CRISPR, and like how does CRISPR allow scientists to edit genes within that setting?
Yeah. So the way it works in the lab research setting, and I'm going to talk strictly from someone who's more translational. There's a
Mhm.
lot of work, a ton of work that gets done on the discovery, the protein engineering side, trying to make these Cas systems more specific, more potent, smaller, right? That's another huge thing here at Mammoth. So all these things are critical to making the system more viable for drug candidate, right? I'm going to talk more about what happens in the academic lab, like if you have a disease of interest you're going after, a target gene or mutation. And I can do a case scenario where let's talk about something I did, Chalk Children's. A disease that I worked on there was Pompe disease. This is a rare pediatric disease that affects the skeletal muscles essentially, and it also enlarges the heart. So without treatment for the severe cases, a lot of the kids with Pompe, unfortunately, they would succumb to the disease usually around 1 to 2 years of age. And as a parent of young children, it's just heartbreaking to hear and see, that side of, the caregiver side. And for a while, there was no treatment. But for Pompe disease, there was something known as enzyme replacement therapy that essentially was approved and treated these children. However, they have to go in every 2 weeks for a dosage, a regular infusion. And it's extremely costly as well, as far as like the insurance side. So this kind of jump started my work in Dr. Raymond Wong's lab, where we were funded by a patient donors grant to really model the system. So first, to kind of think about how to have CRISPR-based technologies correct the mutation. A lot of these rare diseases, they don't even have a cell model or a mouse model to work with. So not only is CRISPR a great tool for therapeutics, it's a great tool for creating your disease model. So that was what I did during my time at CHOP Children's. We created the disease models with point mutations for a few rare diseases like Pompe disease, the MPS diseases. These are lysosomal storage disorders, just to give the rare patients and their families a starting point, right? Where we test before we test these technologies in their kids. We want to make sure the technology works, is effective, and is safe in cells and then in mice before moving on to the patients in clinical trials. Yeah, so that's usually how it starts. We create the model, then we test the different tools and technologies to see how well you are, the efficiency of correcting the patient's mutations, and then looking at all the endpoints to see, hey, does the enzyme for Pompe come back so that these patients can now recycle the harmful glycogen that's causing their muscle dystrophy and their heart enlargement. So that's usually how it works in the academic lab. We want to create the model, cells in a dish, mice, prove the technology's effective and safe. And then usually that research goes on to an industry partner that can scale up that work, right?
Yeah, I was going to ask about that because I have a lot of experience with like sponsored initiated studies, right? But like what does that look like when it's the PI is the one that has sort of this idea and starts,
Right.
implementing it, and so interesting. So you connect with companies that may be able to fund this, right? Is that what you said?
Yeah, and a lot of times it's the patient advocacy groups that
Mm.
spearhead these kind of collaborative efforts where, you know, that technology is taken from an academic setting and translated into the industry setting. A lot of times it's also the proprietary technologies behind these. A lot of academic researchers, we work with the freely available tools, right? And these are a great starting point. But partnering with an industry company and industry in this genome editing field, usually there's evolved variants of either on the nuclease side or on the guide RNA side that can really help you think about packaging this all into delivering into the cells of interest. So yeah, it's a long road from academia, like one
Yeah.
bench scientist who's doing their dissertation work for their doctorate, all the way to clinical trials. But that's how it starts, at least for new technology and growing technology.
Yeah. Yeah, very interesting. I think I'm sure because it's also rare disease, it's also difficult to get patients as well, I would think, right? So,
That's right.
um,
Yeah.
I remember so I'm a big fan of Dr. Wong, and I actually reached out to
Likewise.
him because I'm wanting to get him on my podcast as well. I think he's
Cool.
wonderful.
Yeah,
Yeah, I
I
think he's
agree.
so great. And so but I'm always reminded, like, honestly, out of all the different types of trials that I've been a part of, rare disease just holds a special place in my heart. And so
Yeah,
I'm like, yeah,
likewise.
yeah, there's something about, these kids are so young and they get affected with these diseases that have no treatment. And it's like, it's heartbreaking, just like you mentioned,
It is.
seeing that, the caregiver, the parents are just coming, as often as like several times a week, to get help for their patient, for their kids. So,
And it's amazing the lengths they go to gain knowledge on this, and sometimes they come in with papers published that, I thought, only the academic researchers, no, they get really deep in because,
Hmm.
these are their kids' lives are on the line. They're so invested in seeing a lot. I think I recall a patient said to me, or a patient's mother said to me, you know, this can't be the end.
Yeah.
There's got to be something we can do. Like this can't be the end of the story. And even if it's not for my daughter, I want to make it so that future patients of this disease have something better.
Yeah.
Because and it's just amazing to just be in the just to hear the human side of it, like
Yeah.
yeah, it's more than just a disease when you talk to the patients and their families.
Oh yeah. Yeah. It impacts their life like no other. And especially, it's even crazier when you have siblings that are affected. So it's not
Oh
just,
gosh,
one
yeah.
kid, but it's their two kids may be affected with it, and it's just crazy.
Yeah.
So when we think about sort of like ethical considerations about CRISPR, what are some things
Yeah.
that come to mind in terms of that?
Yeah. So we're a little bit in uncharted territory here. The first approval happened just late last year for a sickle cell anemia treatment. Well, it was a CRISPR Therapeutics Vertex drug known as Exacel, EXA-CEL. It's a shortened name for the actual drug. But essentially, what it does is it is able to use CRISPR engineered cells. We call this ex vivo or outside the body. You can take blood cells, re engineer them so they go from that sickle cell form to a healthy form of a red blood cell. So, and then infuse it back into patients. So, this technology makes sense because you can apply your technology to cells that are outside the human body and infuse them back in. It's autologous, there's no immune response there, right? So the guidance there was from the FDA. There are some guidelines now that a lot of genome editing companies are using as the blueprint as far as off-target cutoff criteria. The big thing about CRISPR Cas9 technologies is we want to be very careful about where this sequence hits across the genome. Sure, you can edit and correct the mutation of interest, but if you hit an oncogene, will you induce cancer, right? So those
Yeah.
are things that are highly, highly important to regulatory agencies. But what are those cutoffs? What are the criteria? Those are still being established, especially as we move from ex vivo outside the body treatment to in vivo. In vivo might be even more strict and more regulated, right? Because
Hmm.
here we're putting the components into the patient's body, and that can have sustained durability of expression. Whereas when you're doing this outside the cells or outside the body in cells on a dish, you can kind of pick the ones that have the corrected mutation and infuse them back. So we're in uncharted territory here, but it's exciting that we're over the first hurdle where the FDA is familiar with these tools, these therapeutic strategies. But yeah, it's exciting to see where it's going to go in the next few years. Yeah.
Definitely. Yeah. Yeah, it's very exciting. So what sort of steps do you need to go from the lab research we're talking about to now into clinical trials? Like,
Hmm.
is I'm sure the FDA requires X amount of patient data and things like that. Are you able to
Yeah.
expand on it?
Yeah, so this also would depend on the disease candidate you're going after. So, like we mentioned earlier, with rare diseases, you're not going to have withhold treatment in a control group. You just don't have enough patients for this, right? For if you're going after a larger patient group, so there is some draw for some companies to go after, larger patient populations, whether it's, type 2 diabetes, hypertension, some things like that on the forefront. there you can start thinking about larger clinical trials. And for a small genome editing startup company, usually these would require partnering with a large pharma company to, enroll patients for a trial of this size. So I'll kind of go to the two extremes, and then you can kind of fill out the middle of, what these would take. So rare disease, essentially, if a early stage startup company or a genome editing company were to go about it, they could develop this type of technology and proof of concept in-house, essentially show that it works in disease models of that rare disease, and have that data package to present to the FDA as an interact type early stage regulatory meeting to kind of line up what do we need to know as far as a safety toxicity profile. And then you go from cells and mice. Same thing you do in academia, actually. It's pretty unique where a lot of people from academia ask what is the difference between academia and industry research. You're still pipetting in a tube. You're still running gels. You're still doing your basic molecular biology downstream assays or biochemical protein readouts. Just a lot of it is more regulated as far as, your documentation. A lot of it is proprietary, so you're not going to conferences all the time, publishing papers on it. Some companies more than other, but it's very goal directed because we have a clear line of sight to get this to the point of pre-IND, investigational new drug, trying to get it towards that phase 1 clinical trial eventually. So in rare disease cases, you have usually phase 1, phase 2 combined together because safety, toxicity, efficacy with limited patient number, you'll see that run together. Just to summarize, it definitely depends on the size of the patient population of that disease indication. So if it's a rare disease, usually a startup early stage company can push it pretty close. With a larger patient population for a more common disease, like I mentioned, hypertension, type 2 diabetes, these would require the partnership of a large pharma biotech partner. Yeah.
Got it. Yeah. It's a lot of collaboration, like it always is in clinical research, right? Like we're having to collaborate with so many different individuals to get
Yeah.
a treatment approved by the FDA, which is like the end goal. So, yeah, a lot of moving parts. So, how does CRISPR technology, how has it been applied in clinical trials for genetic diseases? I know we spoke a little bit about sickle cell anemia that's been recently approved. What are
Mhm.
other diseases out there that are currently this is being investigated for?
Yeah. So actually, a lot of early proof of concept clinical trials happen in the rare disease field. So there's
Mm,
a set of diseases that actually Dr. Huang is very familiar with, known as mucopolysaccharidosis. So a mouthful there. So we call it MPSs. So these MPS disorders, usually they occur in patients very young in age, so pediatric patient population. And they're due to the accumulation of these substrates, we call them, they're known as GAGs, or long word, glycosaminoglycan, or glycosylated aminoglycans, right? So when these build up, the patients have developmental deficiencies in being able to remove this substrate from their bodies. And this leads to all sorts of complications, whether it's enlargement of the liver. You can also have skeletal bone abnormalities. We call these coarse facial features, where actually there's buildup of these substrates in the tissue, in the skin, just all throughout the body. And some of them also have brain a specific CNS or brain component to it, where it affects neurodevelopment, cognitive development, behavioral development. So the reason why a lot of early clinical trials have gone after these rare diseases is there's a very clear clinical endpoint or readout. Like I mentioned, these gags, you can detect them very readily when you draw a patient's blood samples. So there's a very clear diagnostic readout if your therapeutic, your genome editing technology is working. You'll see the levels of those gags or these substrates decrease, which is usually a sign that, you can improve the patient's symptoms over time. Mhm.
Yeah, that's awesome. So, what are some challenges in designing clinical trials for CRISPR-based therapies?
Yeah, so I'd say the challenges for designing clinical trials, a lot of them when I'm thinking about a specific mutation to go after, a lot of it is more in the molecular diagnosis of the patient's genomic sequence, right? So you want to find patients that have that correct or the mutation of interest, right? So, for example, a lot of these diseases, I'm just going to go into a neurodegenerative disease because that'll bring me back to my neuroscience emphasis. So, for Parkinson's disease, it's a very large field, right? I believe 90% of patients have a sporadic form, which means there's not a clear genetic linkage. So, right there, that would rule out a huge chunk of patients because there isn't a very clear mutation that your drug is developed for in that patient. So, that whittles your number down from a large number of patients down to 10% that have familial forms. So, familial forms mean there's a very clear gene mutation that's linked to the pathology. So, and there's a few genes that are usually highly prevalent for something like Parkinson's. So that kind of reduces your number again. So the challenge there is really stratifying your patient population to find the correct patient population based on their disease mutation. And there's something called SNPs too. These are single nucleotide polymorphisms that are naturally occurring sometimes between you and me. We probably have a lot of these SNPs. They're not disease associated, but it's usually has to do with your your genetic background, your ethnicity, right? These are just naturally occurring. So that's what makes it challenging, trying to find that target population to recruit for clinical trials.
Yeah, definitely. Are there any, are there any hesitations that the FDA may have in terms of approving these type of treatments? Like,
Hmm.
are there chances or are there risks to potentially changing something that we wouldn't want to change and things like that?
Yeah, I would say if, you know, previous history in the gene therapy field is any foreshadowing of what's to come, there are, extra sets of guidelines and regulations for the gene therapy field based on, there has been a handful of unfortunate cases where patients have passed away due to more the gene therapy side of it, the components of it, and that's kind of, for lack of better words, set the field back a little bit. So gene therapy, and let me kind of tease apart the two. So I think sometimes in the general public's eyes, you think of gene therapy and genome editing as the same, right? But gene therapy is more an umbrella term for anything like a genomic medicine, anything that gets at your core DNA, your gene sequence, right? So that can entail overexpressing a gene, right? That's known as the traditional forms of gene therapy or gene replacement, where you provide the gene sequence of a gene that's deficient or dysfunctional or mutated in patients. And it essentially replaces your body's ability to make that diseased gene's protein product. Okay. So genome editing is different because here we're usually using the same delivery vehicle. These are viral vectors that are stripped of everything bad inside. And then you put in your good payload, the good technology, your CRISPR components into the middle of this viral vector, and then you can deliver it into the bodies of human patients. So think of genome editing as a subcategory of gene therapy as a field. Yeah.
Gotcha. So you're saying there's less risk with CRISPR because it is more targeted.
I would say, yeah, it'd be actually the flip. There would be more risk because there you're going after, essentially these are molecular scissors that could cut across the genome. But it's also a double-sided coin where in a traditional gene therapy setting where you're overexpressing a gene, you have to look at the toxicity side. If you deliver too much of a gene above normal levels, could that lead to hepatotoxicity, which means liver toxicity, or other sort of symptoms? And I kind of think of this as, I think we're all familiar with the Goldilocks story, where you have to get, that just right in the middle. So usually patients have too little of the enzyme causing the disease. You have to deliver it right to the critical therapeutic levels. If you go beyond that, that's where safety tox studies come in as a gene therapy. So usually in genome editing, we don't have to worry about the toxic levels. We're more concerned about a different flavor, off-target profiles of where our gene editing components are going to hit when it's not on target. On target meaning your disease sequence of that gene. That's what we want. That's the good event. Now we have to scan the rest of your own personal genome to make sure it's not hitting something like I mentioned, like an oncogene, a cancer, potentially a cancer-related gene.
Got it. Yeah. So, yeah, it sounds like the level of risk is just different.
It's different.
Yeah.
That's probably the better word to put. Mm-hmm.
Yeah. So, what does the FDA typically evaluate for CRISPR technology in clinical use?
Yeah, so the first phase is always safety toxicity. So we consider this non-clinical, where we're not yet in clinical trials. Usually what happens here is you deliver your drug candidate in this meaning you have the CRISPR Cas enzyme, the nuclease that does the effective cutting, and your unique sequence that's going after the gene of interest. So you deliver these two. This is following preclinical efficacy. You've shown it works in mice. The next step is to deliver this into non-human primates, or a monkey model. And this monkey model can be wild type, just normal. It doesn't have to be a disease model of non-human primates. And usually what this allows you to see, because NHPs or non-human primate, their genetic sequence is very similar to humans, we can get a gauge of, does this drug candidate target other parts of the genome that we don't want it to hit? So that if it passes that test, where you pass that safety toxicity cutoffs, then you start getting into talks of moving on to a phase 1-2 clinical trial for this drug candidate, genome editing drug candidate.
Cool. And that's what so you mentioned that a little bit in the academic side, right? So that's sort of what you're doing in the lab research before you start, getting approvals to put it into humans.
Yeah, so on the academic side or the early discovery side, work I've done not just here at Mammoth, but other companies and even in at Chalk. A lot of times when you're designing these CRISPR guides, you're using your bioinformatics, your computational understandings. Like you can start to screen out a lot of guides based on computational predictions. So there's a lot of power in leveraging the bioinformatics, you can call it machine learning, AI type fields, to start to filter down the list of guides, the guide RNAs that you're thinking about moving on to the next stage of development, just by looking at there's a wealth of information in bioinformatics where we have genome sequences readily available where we can see with this guide RNA, is it predicted to hit other parts of the genome, right? And try to find one that is not predicted to hit other parts of the genome. And then you have to then where rubber meets the road is you test it in the cells and in mice and then confirm that you're not getting any off-target hits by next-gen sequencing. So, yeah.
Got it.
Mhm.
So, how recent has this machine learning and AI been sort of included in how you go about lab research? I think, for the general public, it's been like such a new thing, but maybe this
Yeah.
is something that's been more, prevalent, I guess, on the academic side for longer. I don't know.
Yeah, I would say it's definitely been a kind of a hot button issue where I think a lot of people are very excited about the applications of machine learning AI. There's a range of emotions. Some people are very realistic in what it can achieve, and some people think it's going to cure everything, right? And be the panacea for every disease. So I take a more measured conservative approach for how we can apply machine learning AI. I'm excited about how it can help us better model future therapeutics. So I kind of think of us in the early stages. We're building data sets, right, on what guides. So a lot of it is theoretical, right? Taking the bioinformatics, the computational models to see how well that matches up with guides we test in cells, in mice, and in NHPs once we get there, and to see how correlated that is. Over time, you start to build up a large data set and you can pipe this in. Are there any patterns, right? Are there any patterns in the genomic sequence that predict for a high correlation between bioinformatic computational modeling to real world editing? And try to establish that line over time with machine learning AI type tools. And that can help guide us to refine and not have to test hundreds, thousands, tens of thousands of guide RNAs so that we can get a drug product out to patients quicker.
Hmm.
Because ultimately, we want to cut down on the time from early research development to clinical trials in the long run.
Yeah. And I was looking at the stats for lab research and before it even gets into humans, and it's pretty high where it doesn't go through, into I think it's like 90% or so of
Yes.
so do you think machine learning and AI could really help and improve on those stats and get things to market quicker?
So I kind of also go with a phrase that things move at the speed of biology ultimately. So there are inherent limitations to how quickly you can get something from, design of your guide RNA for genome editing to the patient. A lot of times you need to build up that data package through testing in disease cells. Cells in a dish can only double and proliferate at a certain rate. Mice, those models take time to breed and inject with your drug candidate. A lot of that does take naturally the time that biology takes. What I'm talking about here is cutting down on kind of the number of test articles you're testing. So a lot of times without prior knowledge of what has a better translatability from we call it in silico, so things we do on the computer for modeling. Over time, we're going to keep learning. It's an iterative cycle where we're going to understand which which type of sequences tend to work really well or which ones perform poorly and don't advance. And those learnings will help speed up the translation, or more than the sheer number of candidates or test articles we have to filter through early research and development. But there is still, it's still, I'm not going to say that's going to cut it down time-wise, like dramatically, but it'll help us reduce the number of, in the end, candidates pools that we start from.
Yeah, I mean, I think there's just so many different layers to how we get treatments to the market, and I think this could be an avenue where it could shorten a little bit, and then there's different avenues like recruitment timelines and things like that
Yeah,
really help overall, just like not make it so it's 10 to 15 years to get treatment to the market, essentially.
agreed. And
Yeah.
on the back end, understanding what is the path to clinical success, what endpoints regulatory agencies are looking for as far as the CRISPR off-target profile, that's all going to shorten the timeline. Of course, this is dictated upon, and this is something we don't know the future of. If it's deemed safe over time, that's going to really change the timeline to development on the back end once you start clinical trials. But with anything like a new technology, I think, there's going to be some test cases out there. My fingers are crossed that the proper due diligence will be done with these companies and the regulatory agencies to ensure that what's going to the patient is not just effective, but primarily the number one thing is safe for the patients and their families. And now I just to kind of see where other, we call them disease modifying therapeutics, have evolved since then. We can look at, small molecule inhibitors. Like usually that has a very clear path. Like if you're taking, this started from aspirin, right? So like things we all take orally, that's a small molecule, a compound. This has had decades of, advancement of multiple, maybe hundreds of thousands of drugs have gone through this pipeline. So it's a very clear, you're looking at pharmacokinetics, pharmacodynamics, your drug clearance over time. So there's very clear markers in clinical trials, what to look for safety and efficacy. So that's on the end, that's a drug class that is much more developed and established. And now we're looking on the other side, CRISPR genome editing systems, where there isn't much established there. It's a new world, and we're going to have to see how it evolves.
Right. It's paving this new path, right, to treatment, and it's a lot of unknown. Sort of like, I mean, there's been examples of, I think it was a physician in China who got in trouble for some of the stuff he was doing, right? So it's sort of like, it's such a fine line, and then there's this hesitation with the general public about like, well, what does this mean with, are we able to sort of have these like designer babies or things like that?
Right.
So yeah, it's definitely a journey.
Yeah. And I can comment a little bit about, some of those fears that from the general public. So that case of the geneticist in China who, and this was all to kind of knock out a gene, CCR5, that is known to allow you to have predisposed some sort of immunity to HIV, AIDS, right? So it's essentially a preventative, a prophylactic approach, which a lot of CRISPR scientists thought we're going too far into, we call it genetic enhancement, right? Something that's not tackling, like on the flip side, we have these rare disease patients that have no therapeutic, it's just palliative care. There's no options, but there's a very clear natural history of where that child's prognosis is going, right? It's 100%, unfortunately, if left untreated. fatal, right? So I think, at least for me, there's a clear line that this type of technology needs to be tested for patients in need rather than, designer babies or genetic enhancement, these type of things. Yeah, and I think a lot of scientists are in alignment with that, where they called a moratorium on research for that at a CRISPR Congress conference following that news. Yeah.
Yeah. Well, I mean, I guess you'll always encounter, individuals who who like this geneticist that you're talking
Like
about.
go rogue, right?
Yeah,
Yeah.
exactly. And
Yeah.
then it scares everyone, and it's just like,
Yeah.
I think the technology is amazing, and there's a lot of hope to be able to treat these patients. So it's
Yeah.
exciting stuff. So, what are the current limitations of CRISPR technology and clinical applications?
Okay. Yeah. So I can speak on this topic for sure. So one of the limitations is when you're not going through like the bloodstream, if you're going to deliver to a diseased tissue, especially I'm thinking of the brain for brain disorders, CNS, you can even think ocular diseases, those of the eye. These require a viral vector for delivery. And there's a packaging size limitation. It's about 4.4 to 4.7 kilobases. Most Cas enzymes are too large. You can't deliver the Cas protein part of your pairing. So we have the CRISPR guide RNA and then the Cas protein that does the cutting, along with promoters that provide that. You kind of think of promoters as the part that churns out the gene of interest. You cannot fit all of that in a traditional, we call these adeno-associated viruses, AAVs, that are deemed to be very safe and have really good tropism, meaning that they can go directly to a certain organ of interest. So that is a huge limitation that holds back some of the technology platforms, because you have to either split up your two pieces into separate viral particles, and that effectively halves your effective dose of drug delivery there. So that's a huge thing. I think delivery due to packaging constraints. And then the second one is the AV field is something a lot of these gene therapy companies are depending on that to advance, because we need that vehicle to deliver the good therapeutic, these gene editing, these gene therapy components to the diseased tissue. So that is a huge, I don't want to call it a bottleneck, but something that a lot of companies need to partner with an AV engineering company to get their payload into patient cells. So the limitations, I think if I were to summarize it in one word, is delivery, delivery, delivery.
Mhm.
Yeah. Yeah.
Yeah. Well, so, these are sort of some bottlenecks right now, but how do you see it evolving in the next like decade or so? And I know a decade might be a long time
Yeah.
for this technology, but yeah, do you want to speak on that a little bit?
Yeah. So if you think about a decade from now, like I mentioned, that Martin Janek et al. Paper in Science that was from the Doudna lab, that was 2012. We're a little past a decade, and it's evolved from something that was kind of a unique tool only done in academic research labs to building out all these startup companies going after inherited diseases, right? Whether they're rare or more common. So I am very optimistic about what the next decade holds. If we've gone from that point to here in 10 plus years, delivery, I think, is something like the AAV components. This is something I've seen just a revolutionary and exponential growth in the investment there. Just because all these gene therapy companies know that a lot of this hinges on getting the drug product to the patient's cells in need. And you can do the ex vivo part outside of the body without these AAV particles, but to get it into patients, you need either AAV or there's something known as LNPs, lipid nanoparticles. And really trying to find that right vehicle is key to unlock the potential of all these technologies. So I'm very hopeful. I think there's a lot of investment in this area to kind of bring together these two critical pieces of the puzzle, where if these evolve exponentially, both of these, we're going to be able to see very soon if we can address patient populations with unmet needs.
Yeah, yeah. And that goes into my next question. How can collaboration between researchers, clinicians shape the future of CRISPR technologies?
Yeah, so this is where I think about my time at Chalk Children's and having an advocate for patients as well as advocate for the bench science behind it. So this is where Dr. Huang is like we talked about, an amazing mentor, and he's kind of that rare physician scientist who can see both sides of the coin and also understands the amount of legwork it takes to go from something that was discovered in an academic lab and that you need to partner with a company that focuses on that technology and bringing it to the patients through clinical trials. And he was always he'd be very proactive in going after, the latest, therapeutic strategy. So I'm thinking of Batten disease, Brineura. That was a breakthrough technology that happened because of his own initiative, his own desire to bring this to his patients that he saw in the exam room. So having those points of contact, the interface, and understanding each other's kind of role and not being wary of each other, I think that's a key thing. Going from academia to industry, there's always some skepticism about each side. And that's changed over time. Like as I've worked in the field, I think the two sides are much more collaborative and willing to work with each other, especially in this new technology space like CRISPR.
Definitely. Definitely. All right, Jeffrey. Well, thank you so much for this amazing conversation. It's so insightful, and I'm excited to even see, where it's at in just a year or two. So maybe we can connect
Likewise.
again to
Yeah.
see any sort of advancements. But yeah, do you have anything else that you want to mention? I know you talked a little bit about fellowships. Maybe you can give some career advice to those that may be interested in lab research, like what
Yeah.
you're doing.
Yeah, I would say don't be afraid to take a chance. I think going to Children's Hospital Orange County was an unorthodox move for a lot of postdocs.
Mm.
Usually they're going on to thinking about being a tenured professor track or going into industry. So it was a move that really broadened my understanding of the field, exposed me to clinical trials. As a bench scientist, I had zero knowledge of clinical trials because all I was concentrated on was, the pipettes, the tubes on my bench and trying to get, data results for a paper, that at the end of the day to fund the research of that academic lab. But working at a children's hospital really give me that sense of purpose, why I wake up in the morning to do the research I do. And it also gave me a portal into the lives of the patients and their families. And in the case of rare pediatric disease, the parents that are so passionate about finding a better form of treatment for their kids. So yeah, that would be my takeaway. There might be, every career path comes with its winding journey, but pursue those passions, try to find out what it is that excites you, gives you passion, and stick with that and be open to new avenues that, even if it's off the beaten track, there's each of these little pieces fits into your career journey. And I'm thankful and appreciative for each step of my career path.
Awesome. All right. Well, thank you. If people are interested in connecting with you, where can they find you?
Yeah, you can. One way is LinkedIn. So there's social media. Another way is, you can, I can provide my personal email. If you have any questions about the topics we covered on this podcast, I'd love to go in more detail or even approach it at a higher level. If this was 80% over your head, I'd love to give you a more higher level overview of it, or just to talk. I love to just get to know people in the field as well.
Awesome.
Yeah.
Okay. Well, we'll link that information in the show notes. All right, Jeffrey. Well, thank you so much.
Thanks for your time, Naomi.
I hope you enjoyed my conversation today with Jeffrey Huang. If you're interested in these types of discussions, please subscribe to our YouTube channel. We also post these episodes on Apple Podcasts and Spotify. For more insights and updates, you can sign up for our newsletter at trialtalksinc.com. Till next time.